Great. Well, first of all, thank you for the invitation to be with you today. I really appreciate the opportunity to talk a bit about some of our work. And I first want to acknowledge that I am thankful to live, work, and play on the traditional territory of many First Nations, including the Mississaugas of the Credit, the Anishinaabe, the Chippewa, the Haudenosaunee, and the Wendat peoples, now known as Toronto. So today I will be speaking to you about some of the community-based research projects our lab has been implicated in regarding oil and gas development and health impacts in Northeastern British Columbia. But briefly, I wanted to first start by just introducing what we do in our lab. So our lab, From Bench to Communities, highlights community knowledge as a valuable source of information in order to explore the relationship between environmental factors and health and well-being. And we pursue the following objectives. First, we develop customized research strategies to identify potential environmental factors associated with health issues observed by community partners. Then, depending on the needs, we may investigate prevalence and incidence of those health issues, or we may develop and use cellular bioassays to study the biological mechanisms that are involved in the development or the progression of those health issues, again, identified by community partners. And all of that together, we hope can contribute to adequate environmental and public health policies by collaborating with regulatory agencies. So before really starting talking about the topic of today's presentation, which is going to be around oil and gas wells, you may already know that Canada is a producer of natural gas, with most of its production being done in Alberta and British Columbia. And just out of curiosity, how many oil and gas wells do you think are present in this region? So you have a couple of choices here. Oh, I hope you can see the polls. There you go. It was on the screen that I was sharing. So I'm seeing a few answers here. That's pretty interesting. I'm gonna give a few more seconds. I think Angela will be the magician of the poll. Perfect. So I see that most of you, 48%, answered around 30,000 wells, and then the second most chosen option was around 10,000 wells. So the answer is actually there's approximately 35,000 oil and gas wells in British Columbia. So most of you got this right. And you can see on this map here a bit of the extent of the development. And you can see that most of them are located in Northeastern British Columbia. We were just able, our group this month, to make the distinction between conventional and unconventional wells. So you can see conventional wells here in the little green triangles and unconventional wells in the little yellow triangles. And then the other colored dots represent different types of industrial facilities. And if you're curious about this process of making those distinctions, we can definitely talk about this during the Q&A. So this region sits on the Montney Formation, which represents, most of British Columbia's production of natural gas. And this represents around 25 to 30% of the natural gas production in Canada. So this is done in mainly Northeastern British Columbia. And we know from scientific literature that there is potential contamination associated with this industry, namely by volatile organic compounds, or VOCs, and also trace and radioactive elements that are naturally occurring in, the shale rock formation, but that can be kind of demobilized during this whole process. So second question that I have for you is, I've been talking just now about making the distinction between conventional and unconventional drilling. For the rest of the presentation, I will mostly talk about unconventional drilling. So just to make sure we're all on the same page, if you could choose what you think is the main difference between the two. So conventional drilling is only used to extract oil, while unconventional is used only for natural gas. That would be option A. Option B would be that unconventional drilling is more simple and cheaper than conventional drilling. And option C would be that while conventional oil and gas resources are developing using vertical wells, unconventional resources require horizontal drilling and multi-stage hydraulic fracturing. Few more seconds, I think. All right, so the vast majority of you got it right. So the answer was C. So when we talk about unconventional drilling, it will be a combination of vertical drilling and then horizontal drilling in the natural gas resource, for example, and that will be used in combination with hydraulic fracturing, which is the injection at high pressure of a fracturing fluid that will create fractures in that rock formation and then deliver the natural gas or oil. So now that we know a bit more what we're talking about, we're going to now start discussing the potential health effects associated with being close to this type of industry. And I first want to talk a bit about toxicological data. So a few years ago, there was a very interesting study that was published by Elliott and colleagues. They evaluated around 1,000 chemicals that have been identified in those hydraulic fracturing fluids, and they screened those chemicals for their potential reproductive and developmental toxicity, evaluated in cellular models, animal models, or also human studies. And what they found is that for 76% of those chemicals, information on reproductive and developmental toxicity was lacking. On the remaining 24% of the chemicals for which they did have toxicological information, 43% of them were developmental toxicants, 40% of them reproductive toxicants, and 17% of them were both. It's important to note that when this study was published in 2016, there was no peer-reviewed exposure assessment studies in the context of unconventional oil and gas in the scientific literature. However, even if there's a lack of toxicological information, for example, there's a multitude of communities observation in the region regarding the impacts of unconventional oil and gas. So people relying, for example, on traditional or wild foods, spending time on the land also are very much aware of potential contamination and the condition of their environment. And so our lab tries to really value local communities' knowledge, experience, and observation, and use that in conjunction with biomonitoring, toxicological, and epidemiological data. That is, that communities' knowledge is an epistemology worthy of respect and that stands on its own. And as such, environmental health projects should seek to target these observations and concerns that were raised or flagged by communities. In 2012, some First Nations reported concerns about the consumption of deer, moose, and elk, which are major food sources in these communities. And specifically, they observed declines in population, but also hunters reported some abnormalities in the animals, such as tumors and growth. And they hypothesized that these may be due to increased contamination related to increased oil and gas activities in the region. They were able to participate in, some discussion, but sometimes the timeline was too short. And so it led to no really validation or analysis of these concerns other than mentioning them kind of as anecdotal events. Although some First Nations, including West Moberly and Saulteaux First Nation, sometimes did their own studies and paid for those studies. So it's in this context that our studies were developed following discussions with First Nations in the region and local healthcare providers. And one of the main areas of concerns we identified during these discussions was regarding exposure assessment. So our populations in this region exposed to contaminants at levels that could be concerning, yes or no. And because the available toxicological data tells us that reproduction and fetal development might be affected by exposure to some of these chemicals, we decided to conduct a pilot study in 2016 by recruiting a small cohort of pregnant women in Northeast British Columbia and measuring their exposure to some environmental contaminants potentially associated with unconventional oil and gas. So this pilot study was led by Marc-André Werner at University of Montreal. It was my postdoctoral supervisor. It was funded by University of Montreal, as well as the West Mobility First Nations. And we worked with 2 medical clinics to recruit participants. And we had a pretty large research team with very involved local partners. So very briefly, the results of that pilot study, which were published in 2017, I think, 2019, showed that one benzene metabolite, so benzene is a volatile organic compound, and to measure exposure to benzene, we can measure some of its metabolites in urine samples, for example. That's what we did. So the levels of one of this metabolite called trans-transmeconic acid, or TTMA, was quite higher in the pilot study participants compared to the general Canadian population. And then when we further looked into levels of TTMA, specifically in the Indigenous participants to the pilot study, the levels were even higher when compared to the general Canadian population. We also measured a variety of trace elements, and we found that in particular for barium, aluminum, strontium, and manganese, levels were quite higher in pilot study participants, both in urine and hair samples, when we compare to reference populations. And again, for barium and strontium, the levels were even higher in Indigenous participants compared to non-Indigenous participants. So this pilot study allowed us to observe higher levels of some contaminants in the participants, but it had several limitations, including the small number of participants and the fact that we did not have measurements in indoor air and tap water, for example. We know that these compounds that we measure, some of them can transfer through the placenta and reach the fetus. So with all of that in mind, we designed a larger biomonitoring initiative to confirm or disconfirm the results from that pilot study. This study is called Exposures in the Peace River Valley, or EXPERVA. So it has a big exposure assessment component, but also we investigate endocrine disrupting activities of environmentally relevant concentrations of contaminants that we measure in the participants, in the EXPERVA study participants, because of the potential reproductive and developmental toxicity of these contaminants that could occur through endocrine disrupting mechanisms. So I'm going to present you some results from EXPERVA, which is still ongoing. So this study is led by Marc-André Werner at University of Montreal and also myself. It is a CIHR-funded project. We also put in place an oversight committee whose mission is to evaluate all the aspects of the research project prior to their use. And once again, we have a pretty large research team with very involved local partners. So the recruitment was conducted in the summer of 2019 in Northeast BC. We worked with medical clinics and a midwifery clinic, and local partners also advertised the project to their members. We recruited a bit more than 85 participants, but 85 really completed the whole thing. And around 90% of the pregnant women that were met to just that we presented the study to decided to participate. So we were quite happy with the success of this recruitment. So there's a lot of different things going on in EXPERVA. There's exposure assessment, there's toxicity assessment in the lab. So it's fairly complicated. This is just the methods, very briefly presented of the exposure assessment objective of this study. So we use a multimedia approach that combines environmental monitoring of VOCs and trace elements in tap water and indoor air, and biomonitoring of trace elements and VOCs metabolites in urine, hair, and nails. So that's first thing. And then we also estimate the oil and gas activity around the participants' residence, and we use 2 methods to do that. The first one is we calculated the density of wells around the participant's home. So that's just the number of wells that are present. And we use different buffer zones around the participants' homes. So a 2.5, a 5, and a 10-kilometer zone. And then the next thing we did is we calculated metrics that consider both the number of wells around a participant's home and the distance of these wells to the participant's home. So these metrics are going to be called in the presentation IDW metrics. That stands for inverse distance weighting approach. That's what you can see here on the screen. So it's a very simple equation where the wells that are the closer to the residents have a bit more weight in the equation. All right, so just briefly some characteristics of the Experva study participants. So the median age of participants was 29 years old at the time of recruitment. Around 20% of them identified as Indigenous. The median gestational age at time of recruitment was 23 weeks pregnant, but it went from, I think, 5 weeks pregnant to 40 weeks pregnant. So we had the participants across trimesters. Most of the participants have had previous pregnancies before this one. 8% reported smoking during their pregnancy, and 23% of them were exposed to secondhand smoke at some point during their pregnancy. Around 20% of them had an attached garage, and the majority of the participants' source of water in the home was city water, and also the majority of the participants either drank filtered tap water or bottled water. So smoking, exposure to secondhand smoke, and also having an attached garage can all contribute to the levels of VOCs measured indoor. And then also the source of water can contribute to the levels of exposure to different contaminants, either in tap water, obviously, but even in indoor air. So that's why we ask that kind of questions to the participants so we can assess other sources of exposure to contaminants. So in total, we identified in 2019 a bit more than 29,000 oil and gas wells in the region at the time of the study. I thought it would be interesting just to show you the kind of maps that we can generate in order to calculate those density and proximity metrics, so those IDW metrics. So the first image here that you see is just all the wells that have been drilled so far in British Columbia. So here would be Alberta, just to situate you a bit better. In the second map, the wells are now shown in pale green, and the purple dots represent the residences of Experva study participants. And then this is just a close-up to show you how the wells can be close to residences. But it's important to note, however, that not all those wells are active. Some may be abandoned or decommissioned for a very long time. Some may be inactive, and then they will all be in different kind of phases of production. In Experva, the median number of wells around the participants' residence in a buffer zone of 10 kilometers was 90 wells, and the maximum was 346 wells. So here's some results. We measured a variety of VOCs in indoor air. And so you can see that on the screen here, the median levels of those VOCs in Experva are shown in the blue-green bars. And what you see in the yellow/orange bars are the median levels of those same VOCs reported in the Canadian Health Measures Survey, or CHMS. I'm assuming most of you are familiar with CHMS, but just to be sure, CHMS provides nationally representing levels of a lot of different things, and that includes VOCs in indoor air. So we analyzed 47 VOCs, and 40 out of those 47 were detected in more than half of the Experva participants, and also had nationally representing levels provided in CHMS. For 27 out of the 40 VOC in indoor air, median levels in Experva were between 1.3 and 3 times higher than the national median levels. And in particular for the VOCs you now see identified by red boxes, the proportion of participants from Experva with indoor air levels of those VOCs over the 95th percentile reported in CHMS ranged between 10 and 28%. So the CHMS 95th percentile for the indoor air level of a specific VOC means that for 5% of their participants, indoor air levels of this VOC will be higher than that threshold that is called the 95th percentile. So if exposure to VOCs in Xperva was similar to general Canadian population, we would also see around 5% of our participants with indoor air VOC levels above that 95th percentile. But what happens is that for a good number of those VOCs, we have more than 5% of participants with indoor air levels above that CHMS 95th percentile. It's also important to note that there's a lot of different sources for those VOCs, including putting gas in your car, using nail polish and so on and so forth. But there is some literature on the presence of those VOCs around unconventional oil and gas development. And some of them are also detected in hydraulic fracturing fluids and the generated wastewaters. So after looking at the levels, we then looked at associations between the density and proximity metrics and those indoor air VOCs. And we didn't do that for all the VOCs. We just selected those with particularly high levels in Xperva. And I'm just going to show you some examples today for acetone, 2-methyl-2-propanol, and chloroform. So we use multiple regression models here. What you can see in the table is the adjusted beta with the confidence interval. What is bold was statistically significant. So the beta is the degree of change in the outcome variable. So in this case, indoor air VOC, for every unit of change in the predictor variable, in this case, either the well density or the IDW metric. And so a beta of zero would mean that there's no relationship between the predictor and the outcome variable. And a positive beta, for example, would mean that an increase in our IDW metric or density of wells would yield to an increase in indoor VOC. So after adjusting for some covariables, we found positive associations between the 10-kilometer buffer well density metric and acetone and chloroform indoor air levels. And we also found similar associations between with the IDW metrics considering all the wells in the province. It's important to note that the indigenous status contributed significantly to the models for acetone and chloroform. And this is further illustrated by the fact that the indoor air levels of those two VOCs were higher in the residences of Indigenous participants compared to non-Indigenous participants. And finally, we also conducted sensitivity analysis in these models. So for example, we included the use of nail polish remover in the week of sampling, if the participant was storing paints and solvents in their home or garage. For the chloroform model, we also included a sensitivity analysis with the source of residential water, as VOCs present in tap water can evaporate and then be found in indoor air. And all the sensitivity analysis we conducted still supported the significant association between those density metrics and indoor air concentrations of acetone and chloroform. So before talking about the last set of results, I have the last poll question. So why do you think we conducted these sensitivity analyses in the model? Is it to identify potential errors in the model, or is it to account for other sources of exposure to VOCs? All right, once again, very good answer. So most of you selected B, which is the reason why we conducted those sensitivity analyses. So it was to account as much as we could for other potential sources of exposure to VOCs indoor. So the pilot study, if I go back, was the catalyst of Xperva, but also some epidemiological study that is ongoing in our research team, where we look at potential associations with birth and maternal outcomes. So this was started when I was a postdoctoral fellow at Université de Montréal. And so based on previous epidemiological literature and toxicological data regarding contaminants associated with this industry, we also wanted to evaluate associations between maternal residential proximity to these wells and birth outcomes, and also maternal outcomes. And we used birth records data from the largest hospital in the region over a 10-year period from 2007 and 2016. So approximately 6,000 births occurred between 2007 and 2016 at that hospital. 58% of the women have had previous pregnancies. 12% of them were reported smoking during their pregnancies. We didn't have information on the gestational age of around 1,000 women, but on the remaining births with that information, 10% of the infants were small for their gestational age, and 4% of them were born prematurely, so before 37 weeks pregnant, 37 weeks pregnancy. So we first evaluated the association between those density and proximity metrics and birth weight. And to do this, we use, again, multiple regression analysis, and we corrected for some covariables, such as number of previous pregnancies, infant sex assigned at birth, mother's age, and smoking. So on the figure, you can see something similar than what I presented before, just in another format. So you see the adjusted beta. Those are the central points with their respective confidence interval for the 3 buffer zones and for each quartiles of exposure. So in this study, when we calculated the IDW metrics for each of the birth, we then separated them into quartiles. The first quartile being kind of the reference group, aka the group that was the least exposed. So after adjustment, we found negative associations that did not match an expected dose-response relationship. So for example, compared to women in the first quartile, women in the second quartile of the 10-kilometer buffer, and women in the second and third quartiles of the 5-kilometer buffer for the density metrics had babies with birth weights between 41 and 47 grams lower than in the reference quartile, but we didn't see any significant effect in the fourth quartile, which is the highest exposed group. In a new study that we recently published, and I should have changed under review, it's now published, we evaluated the association between those same metrics and mental illness and substance use among pregnant women in this data set. And so what we found, first on the left, you can see the results of these models for a 10-kilometer buffer, and on the right, we used a larger buffer of 50-kilometer, and we can talk about that if that's of any interest during the Q&A. So first, looking at the left graph, what we can see is in the second and the third quartiles of density proximity metrics in that 10-kilometer buffer zone, we observe a positive association with depression compared to the first quartile. And when using the larger buffer zone, the third and fourth quartiles was associated with increased depression, anxiety, and substance use. So to conclude, I think I'll be right on time. We do have some epidemiological and toxicological literature that suggests risk for poor birth outcome and maternal outcomes, including in Northeastern British Columbia. I didn't present some of the epidemiological literature from other regions in the world, but there is quite a getting larger, but a body of studies on that. Most of them have been conducted in the US, and the vast majority of them find associations with poor birth outcomes as well. We still face an important lack of data on exposure assessment and mechanisms of toxicity, including in the context of fetal development. And hopefully Xperva can help a tiny bit bridge that gap, including on mechanisms of toxicity. And I'm happy to talk about this during the Q&A if that's of any interest to some of you. In Xperva, we do have some preliminary evidence of higher exposure levels to some VOCs and also trace elements. I didn't have the time to present this data, but we do have also similar results than in the pilot study regarding trace elements. We also have some preliminary evidence that density and proximity of those wells can potentially contribute to residential levels of VOCs and also trace elements. That tells us that using this kind of metrics, although it has a lot of limitation, because it's based on localization and doesn't account for wind speed and direction, for example, can still maybe be an acceptable estimation of exposure strategy, at least for certain VOCs. And I want to take a few seconds just to thank the amazing work of all the team members in Experva and our local partner, especially West Moberly, Saulteaux First Nation, and the Treaty 8 Tribal Association for welcoming us and doing this work with us. And thank you also for your attention, and happy to take any questions.